Welcome to the Lactase Prelab video. In this particular video, we'll cover the expected learning outcomes for the lab, review your Prelab assignment work, talk about an experimental overview of this particular lab, and end with your Postlab assignment work. The expected learning outcomes include the proper use of pipettes and spectrophotometer, understanding of experimental design as relates to this particular lab. We'll review the appropriate data analysis, in this case that includes graphing, do an introduction to reading scientific literature, writing a complete lab report, and understanding of enzyme structure and function. For the pre-lab assignment, you should read the Lactase handout, view the methods videos on pipetting technique and the use of a spectrophotometer, view the protocol video, and take the pre-lab quiz. Following that, you'll actually perform the lab, which brings us to the experimental overview. We need to focus on lactose and lactase. It turns out lactose is a disaccharide composed of glucose and galactose joined by a glycosidic linkage. Lactase is an enzyme, a protein, which hydrolyzes that glycosidic linkage and produces two products, galactose and glucose. Now this is important for us in that the former, i.e. lactose, cannot be digested in our gut, where galactose and glucose can. So it allows us to access these energetic resources. So this lab is going to focus on the lactase enzyme, which, as I said, converts lactose into glucose and galactose. But we have a problem with this particular reaction, in that neither the reactant or the products absorb light in a wavelength that we can monitor. However, we have a solution. And that solution is a synthetic lactose analog substrate called OnpG. Now OnpG is reacted on by lactase to produce two products. It cleaves the glycosidic linkage, and those happen to be galactose and another important product called O-nitrophenol, which happens to be bright yellow. And this is very advantageous in that it absorbs light at a wavelength of 420 nanometers. So we can monitor this formation of this product in a spectrophotometer, which indirectly allows us to determine lactase activity. In addition to this, we've talked about earlier how enzymes are affected by their environment. And one of the relationships we covered was that of substrate concentration. And we know this Michaelis-Menten saturation curve shows that relationship. So what we're going to do is we're going to expose our lactase enzyme to various concentrations of the substrate and see what happens. In addition to this, we also know that enzymes are affected by their environmental parameters such as temperature and pH. And here again, we're going to conduct two other experiments in which we vary the temperature and the pH of the solution and monitor enzyme activity. We're going to do this at a variety of concentrations, and you should be able to determine by the amount of yellow product the optimal temperature and pH conditions for this particular enzyme. Once the lab is completed, you're going to move on to your post-lab assignments, which include viewing data analysis videos on graphing fundamentals and how to generate a scatter plot. This will be very useful in generating your report. To that end, you'll also read the lab report assignment sheet, which will walk you through the step-by-step instructions for that report. You'll also review the online reference papers, and once you have all that, all you have left to do is write your lab report. So enjoy!